We have 36 hours.
Okay, guys, watch your ears, everybody. Welcome to Wednesday Yachting Ocean. Live from the St. Francis Yacht Club as we work through all the technology of being able to live stream and record each of our talks. So you want to come by next week because we've all heard of J105s. It's one of the most popular one design classes on the planet. And the designer of J105s, as well as the international president, Nicole Brolt, Rod Johnstone, the designer, and Nicole Brolt, and a former president of the J105 class, Bruce Stone, will all be here to give us a talk all about the colorful beginnings and the rapid growth of this incredibly popular one design boat that is literally around all over the country and continuing to grow. Then, of course, the next week after that, on October 30, we'll be able to listen to Amy Bridge. Amy Bridge is the U.S. representative of the Neptune Project. The Neptune Project is fascinating. It's an organization that is taking people with Parkinson's and giving them their kind of dream to go sailing. And not just sailing, but to compete in parts of the around the world race. So this is quite an ambitious, wonderful project. And Amy Bridge will be here to tell us all about it and give us examples of people who in their dying moments have gotten to achieve something that was otherwise just a dream. And we've all been watching SailGP develop as an event, and it is maturing as an event. And when we see these big audiences watching SailGP and or the America's Cup, we're paying attention to the yacht racing, but something else is happening. That is to say, an architect and a team are creating these on-site venues for spectators. Their view is that spectating can come to sailboat racing, but only if you design the venues and the arenas and the courses just right. Well, the person who knows most about that is Chief of Global Architecture for SailGP and the America's Cup, our own Chip Nielsen. Chip was an architect with great credentials who took an opportunity to combine his interest in sailing and his knowledge in architecture to become this important person in the America's Cup scene. So he'll be here on November the 6th to tell us all about his game. I got a little bit of sound here, Mark. I don't know what that's all about. A little bit about our speaker today. So how did we get here? What caused human beings to evolve atop the animal kingdom? What were the biological branches that caused these little microbes to grow into multi-cell beings and ultimately into intelligent beings who could, for instance, give talks like today? Well, our speaker today is very qualified to speak about this. He's got a genuine British accent. I asked earlier, is that a real genuine accent or just one to impress us? It's real. He was born in the UK, and his first ventures on a boat were simply taking ferry rides to France, which is a fun thing. I've done that too. Now there's cool trains you can do it with, but either way, it's fun to go over to the mainland from Britainland. At age 18, he found himself on a dive boat off the shore of Madagascar and becoming incredibly fascinated by the biodiversity that he saw in the water. He went on to the University of Sussex to get a BS in marine biology, and then to SUNY Stonybrook to get a PhD in marine biology, and specifically on the evolutionary development in the biosphere that we see as the aquatic environment. He did postdoc at UC Berkeley, and then went on to be a professor at University of Chicago. And during the course of his career has now got 1.5 million miles of ocean exploration under his belt. Currently, and since 2019, a professor at Stanford, he's basically studying the developmental biology and the biomedical tools that are evolving rapidly, not just in Silicon Valley, but elsewhere, that allow for the exploration of this evolutionary path that is allowing such biodiversity. He has an interesting premise. He said, thus far, the study we've been doing on animals has been chosen by just those animals we could easily get our hands on and which we think have some relationship to humans. He suggests that in the vast biosphere in the oceans, we have the opportunity of far more exploration of biodiversity. There's more there, and it's readily available for us to study. So in order to talk about the major changes that caused us to evolve to the sentient beings that we are today, we couldn't have a better speaker than our professor from UC Stanford, Chris Lowe. Chris, tell us all about it.
Okay, well, for those of you who are confused about you've come to see sharks, there happened to be another Chris Lowe from Southern California who actually I get phone calls for all the time, who works on sharks. But here we are today to talk a little bit about biodiversity. So this is where I work. This is the biology Hopkins Marine Station, Stanford University, right next to the Monterey Bay Aquarium. And one of the reasons I'm there is because I'm interested in evolution of animals. All life originated and diversified in the ocean. Everything started out in the ocean. And so the ocean really, if you're interested in biodiversity, provides this unparalleled opportunity and access to that biodiversity. And marine stations specifically, we don't have that many marine stations attached to major universities. But marine stations are essentially a portal for access to biodiversity. And if you're a biologist interested in the role that biodiversity plays in our fundamental understanding of animal biology, then marine biology stations are really a central focus. All these animals that we see on the board here are not the ones we typically think about when we think about biomedical research and the role that molecular biology plays in our understanding of animal biology. But what I'm going to be talking about today is it is this kind of opportunity that biomedical research can now be applied to really mine and to find out fundamental principles of biology using more than the usual suite of animals that we typically study. And so any biomedical research that you're familiar with, anything that's related to cancer biology, to regenerative biology, basically most mechanistic biological science has been based on studies on these kinds of animals. And so there's a group of animals that people are very familiar with studying, which is very close related to us, the vertebrates. So mouse is the main sort of lab rat that we use for understanding mammalian biology. But zebrafish is another incredible system, along with Xenopus laevis, the frog. And then there's all sorts of really interesting fundamental science that's come out of Drosophila melanogaster, the fruit fly, and also C. elegans, the nematode. So these animals have been picked, they've been mined in great molecular detail, and they provide us with an incredible insight into the mechanisms of how animals work and how they go through dysfunction. However, if you look at where those animals sit on the tree of life, so on the left-hand side, you can see a tree-like structure, which is basically representing biodiversity writ large. So you can see all these different lineages that are in brown, and you can see the different model species that are placed on that tree. And you can see that there's a bunch clustered in the vertebrates, and then the nematode and the fly are positioned at the other end of the biodiversity. But just by a cursory glance at that, you can see that the classic model species that we tend to study are really a minority of the biodiversity that exists. So if our goals are to really understand biology writ large, then there's this extraordinary range of biodiversity that we have not accessed from mechanistic studies and are an incredible opportunity to understand biology. Each one of those, do we have a little pointer? Do you have a pointer? Ron, did you have a pointer anywhere? No worries. So each of these brown lines that lead up to the species that represent 1 billion years of independent evolution, these are natural experiments in animal biology. Each individual lineage, which leads to a phylum, so a phylum being a mollusk or a worm or an arthropod, animals have spent almost 1 billion years of independent evolution solving basic biological problems. So they represent these evolutionary experiments which are accessible to us by mining the extant biodiversity to understand how animals have evolved and adapted to the complexities and the challenges of life. many years ago, well, not that many years ago, marine biodiversity, before the advent of model species, really were some of the foundation principles of some of the major discoveries in biomedical science. So the jellyfish Aporia victoria, from studies that were started at Friday Harbor Labs, was the species that was mined to discover green fluorescent protein, which is now a fundamental technique that's used for labeling up cells in living animals and forms the basis of many, many neurobiological studies and many, many insights into how cells function in animals. Aplysia californicus, which is the sea slug, was the mollusk that Eric Kandel, a famous neurobiologist who won the Nobel Prize in 2000, it was mining the biology of the sea slug that gave some guiding principles into how animals learn. So there has been a precedent for using biodiversity in cell molecular biology, but this changed pretty significantly in the 80s. As model species took off, as DNA sequencing became within the reach of new model species, and DNA cloning was used gene cloning was used very commonly, the kind of techniques and the kind of expense that was involved in mining biodiversity was really restricted to these model species. It was simply not possible and out of reach in terms of expense for applying these new tools to diverse groups of animals. So research communities really focused on individual species representing particularly mammalian development, but also in fruit fly and in nematodes. And these species were often picked because of their amenability for being raised in the lab, rather than for the representation of animal biodiversity. That equation has now changed pretty significantly. Over the past even 10 or 5 years, the cost of genome sequencing and the advent of genome editing, CRISPR-Cas9 technology, where you can go into a genome and you can actually manipulate gene function. That tool set is now applicable to pretty much any animal that you could pick out of the intertidal. And so the rationale for mining the biodiversity that we see in our oceans has suddenly become, first of all, cheaper, and also within the reach of individual labs to make an impact by picking an animal with interesting biology, with interesting biological properties. So this is to bring us back to the marine biology stations again. If you want to access that biodiversity, and as I said, marine biodiversity by far and away represents the most amazing biodiversity available to us on the planet, then marine stations occupy a key niche in institutional biology for accessing that biodiversity. So my contributions and my lab's contributions to the use of biological mechanistic tools to address fundamental questions of biology has been based on these rather weird-looking animals. Unlike the classic animals that you're used to seeing, the starfish here on the right-hand side, which I'll tell you quite a lot about today, and then also this rather unusual-looking worm called chordates, are animals with really interesting biological properties, but they are not the charismatic animals that you generally think of when we hear a marine biology talk. But they have interesting features of their biology, which makes them an opportunity for exploring how evolution has solved various problems. So I'm going to be talking about body plans, about how animals are organized, and the major differences between the groups of animals you're familiar with, what's different between a fly and a vertebrate, or between a mollusk and a worm. That's based on their organization and what we call a body plan. And so a good comparison to what we would be more familiar to what a body plan might be is if you look at the chassis of a car. You have the four wheels and you have the engine at the front and you have the chassis. This has been adapted in a variety of different conformations in the evolution of vehicles. So we start off with our very basic car, which we go on to our Lamborghini. It's been further modified into a variety of different forms, all the way to buses and dumper trucks, and then is even recognizable in the formation of aviation. So the body plan is the basic organization of the major groups of animals, which are represented by 32 phyla in the animal kingdom. A more familiar example is our own phylum, which is called the chordates, which includes vertebrates, but also more simple animals. Here is in its simplest form, the chordate's simplest incarnation is an animal called Amphioxus, which has a dorsal nerve cord and a brain, which is shown in blue that runs along the dorsal surface there. It has perforated gill slits, which we see in fish in the anterior part of the gut. And then they have segmented muscle blocks called somites around the whole body. And every single chordate that you see is characterized by these basic features, the basic body plan. So here we have a hagfish feeding on a whale carcass off the Monterey Bay. Here we have our familiar fish, and then all the way to mammals. So this basic body plan is what defines this group of animals, and it is that kind of organization that I'm interested in, and how that changes over evolution. Another principle which is a fundamental part of a body plan is an axis. Once you have multicellularity and you have many cells in an animal, the way you specialize those cells is to organize them along organizational axes. So the one that's most obvious here is what we call the antero-posterior axis, the head to tail axis. And you can see anything from a snail and a fly all the way to a shark are organized along an antero-posterior axis. So the most anterior is organized around the head, often defined by complex sensory and processing centers such as the brain and the eyes and the sensory modalities. And then all the way back in the more posterior parts of the animal, which is defined by the trunk and the tail. Every bilaterian animal, an animal that most animals we're familiar with, are defined by an antero-posterior axis. Also, the dorso-ventral axis from the back to the front. And that's apparent even in animals a lot more unfamiliar. These bilaterian animals, the hemichordate here and the phoronid animal down at the bottom here, these are all characterized by similar axial organization. They have a head and a tail axis. And it's that head-tail axis I'll be talking to you a little about today. A fundamental part of studying and understanding how axes change in evolution is how they're formed and how they change. And this happens during the process of development. Developmental biology is the process where you go from a fertilized egg all the way to a fully formed animal. This is a movie showing 24 hours in the development of a fish, a zebrafish embryo. You can see here it's sitting on a yolk sac, and those little cells that are dividing are the cells of the embryo. So you have initially cell division, which you can see is producing a little hat of cells which then start to move and envelop the yolk. You can see on the top the head forming, and then the tail is forming all the way around the edge of the yolk sac here. So that process of developmental biology is where all the fundamental features of the adult body plan of all animals is starting to form. So any changes that evolution enacts on an animal, any sort of morphological change that happens in evolution has to happen during the process of development. It's when all the major features of the animal, the axes, the morphology that define the animal are formed. They all are unfolding during the process of developmental biology. We're more familiar with something like a fish. You can see basically a little fish tadpole that's forming in the top left-hand panel. A less familiar one are these sea urchins. So a sea urchin actually has an early larval form, which is bilaterally symmetrical. And this is a process of metamorphosis from the larva to the adult sea urchin that's happening over a period of 30 minutes. You can see the little spines that are sticking out of the side of the larva. And you will start to see the little tube feet poking out of the side of the larva, and the animal walks away. So not only are the adults themselves fascinating and beautiful animals, but the developmental biology, which is microscopic, which is happening in the ocean column at any particular time as you're sailing around the world, it is just as diverse and just as beautiful as the adults that they give rise to in later stages of development. So one thing over the past 40 years that's come out of comparative cellular and developmental biology is that despite the difference that we see in the morphologies that define a vertebrate like a shark and a arthropod like a fly, they have quite different morphologies, but the underlying principles, the underlying genes that define the head from the tail are the exact same genes. So even we have this amazing diversity of morphology, the underlying molecular biology and the genes that define the major territories of the animal body plan, particularly the head to the tail axis, are highly conservative. So there's this rather enigmatic observation that this amazing biodiversity that we see in the animal kingdom is actually underlain by fundamental molecular conservatism. And so I'm really interested in how this molecular conservatism has been applied, not just in the animals that look more familiar to us, but in more unusual animals. How do they use this molecular anatomy to define their more unusual body plans? So the way these genes were discovered were back in the 1960s. A guy called Ed Lewis in Caltech discovered these rather unusual looking fruit flies that had major transformations in their body plan, particularly along the anteroposterior axis. So here you can see on the left-hand side is a fly head showing its normal mandible structure. And here you can see a genetic mutation in one of these genes that define the apiaxis of an animal. And you can see now that the antennae have been replaced by legs. So there's been a transformation in the head segments from antennae to legs. So there's basically structures that are normally in the thorax of the animal are now being expressed in the head of the animal. In another animal, you can see the normal morphology of a fly is just two wings with two balancer organs. In this mutation, you can see that they now have two pairs of wings. There has been a major transformation in the organization of the anteroposterior axis of this animal. And what they found is that there's a whole suite of these genes that are molecular switches that turn on other genes on and off during the development of an animal that are involved in regionalization of the anteroposterior axis of the animal. And what was rapidly discovered after this insight from Drosophila melanogaster is the exact same genes that are involved in specifying different regions of the arthropod body plan. The same genes are deployed in a very similar way during the development of the vertebrate body plan and are also involved in regionalization of the vertebral column and the central nervous system of animals. So here you can see this beautiful book by Neil Shubin. If you just look at color coding, the genes themselves are not that important. It's more where the genes are expressed. You can see that the red genes, which are involved in formation of the anterior part of the fly, are expressed at the base of the brain of a vertebrate. And the nested expression of these genes down the axis of the animal are functionally related to their role in regionalization of the body plan. So, along with the vertebral column and the spinal cord, they also found a whole set of genes that have conserved roles in the formation of the fly brain and the formation of the vertebrate brain. So the same genes that specify the front and thinking part of the fly brain, those are the exact same genes that are activated and involved in specification of the forebrain of the vertebrate. And so we find, despite this molecular this morphological disparity between all these different animal groups, the basic principles of how you define the front end from the back end of an animal, and particularly in the patterning of the central nervous system, these genes are highly conserved during the development of these animal groups. So the way I like to describe this suite of genes is a type of hidden anatomy. It's a molecular anatomy that we have to use tools to actually reveal what it looks like. But it's a certain type of anatomy that is invisible to us as observers, but takes molecular biology to reveal how they're expressed. So the principle that in our labs that we've been thinking about is how can we use this concept of molecular anatomy to explore the more unusual body plans that we understand very little about? Can we use these tool sets deployed in a new animal where we don't understand how they're organized? If we look at their expression and their role during the development of these animals, this gives an insight into how those animals have evolved. And so the two groups that we've been interested in the lab at looking at is the echinoderms, represented here by starfish, and then also this group called Entropneus that I won't talk too much about today. But I think if we move on to the next slide, I think that most of us if you look at a fly and you look at a human, if you're told to have an exercise like where are the equivalent regions of a fly and a human, most of you would be pretty capable of defining where the head and the trunk is in the fly. And Hollywood has done a very good job of comparing what happens if you mix the DNA of a human and the fly, then you get something that looks pretty much like this. So this is not a challenge from a morphological perspective. But I would argue, I would challenge any of you to come up with a comparison here. How would you take the body plan of Ron and compare him to a starfish? I think the concept is, where is the head and the tail of the starfish? And it sounds like this is a very familiar secret here. Everyone is familiar with the starfish, but these animals are closely related to us. These are one of the most closely related invertebrate groups to our phylum. And yet they have remained this biological mystery for 150, 200 years of zoological study. One of the most complete sets of invertebrate textbooks by Libby Hyman, who was probably the preeminent zoologist in the last century. She basically, her phrase that I love is, "I hereby salute the echinoderms as a noble group, especially designed to puzzle the zoologists." And it's true. To this day, we don't know how to relate the echinoderm adult to its closest related groups. No. We'll go through the various iterations, and you can vote on which one you like the best. So it's not just at the level of their weird anatomy that they're different. The fundamental difference is that these groups belong within the bilaterian animals, but they have completely lost bilateral symmetry and become secondarily radially symmetrical. So it is very difficult to identify where the head and the tail of a starfish is based on pure morphology. What our thesis is that the work that we've done in heavy chordates, but also work from chordates, shows this beautiful hidden molecular anatomy of genes that mark the head and genes that mark the tail. So the simple question is, if we clone those genes and we examine what the genes are doing in an echinoderm during its development, will this give us a clue into how this animal is fundamentally organized? Classically, what we do before we get into that kind of study is look at fossils. Is there a fossil record that explains the transition from bilateral symmetry to pentaradial symmetry? And there's an exquisite fossil record for echinoderms, but the intermediate forms are so dramatically different that it's been impossible to really reconstruct how echinoderms evolved. The puzzling thing about development of starfish and echinoderms is they start off as perfect respectable bilaterian animals. These are starfish larvae with a clear anteroposteral axis and a clear dorsoventral axis. Midway through larval development, they start to develop a little starfish on the posterior end. So you can see that sort of dense structure around the trailing end of the animal. That is the developing starfish on the end of the stomach of the developing larva. And after metamorphosis, suddenly the starfish emerges from the larva. It's a very radical and violent process of metamorphosis. So this is a project that I had done with Dan, who's been a long-term collaborator of mine. He gave a talk here about a year ago, and a very talented postdoc that we shared together, Laurent Formery. In collaboration with a company here in the Bay Area, Pacific Biosciences, is a long-read sequencing company that we collaborated with on this project. So here's the models. So here's the models that you were talking about, the competing models of how people have thought about echinoderm evolution over the years. So one is the bifurcation hypothesis. That is basically summarized by the Patrick starfish from SpongeBob. One is the circularization hypothesis, which is the idea that the starfish head joined the starfish tail, and then each arm of the starfish represents a different region of the apiaxis. The duplication hypothesis is probably the one that's most well known, which is the idea is that bilaterians joined at the head and duplicated 5 times. So the middle of a starfish represents the joining of a head, and each end of an arm represents the posterior end of an axis. And that's nicely demonstrated here by my colleagues, showing heads joined at the middle and tails along the outside. And the other one is that the mouth to anus axis is the apiaxis. So the end of the starfish you look at when you look down on the starfish, where the anus comes out, that is the posterior, and the anterior is where the mouth is. Those are the hypotheses that have been proposed. So I'm going to briefly tell you about the approaches that we took to address these questions. One is a more precise one is a more unbiased and a broad technique. So the first one that I'll mention briefly, but not in great detail, is RNA tomography. So the way we do this is we take a little baby starfish, we cut out each arm like a pizza slice, and then we embed those arms and we section in great detail down in 3 different dimensions. And each section we sequence and identify all of the genes that are expressed in that particular segment. And then we look for the genes that are markers of the head and the tail axis of the starfish. So one is RNA tomography, and the other one I'll tell you about, which is a more precise one, which is really looking at, on a cell-by-cell basis in an animal, where a gene is expressed. So the first one, RNA tomography, here is a model showing what we did, which is basically to take an arm, and we section through and sequence each one of those slices, clone the genes, and look to see what those genes were and where they were expressed in the animal. And what we found is that there was very little support for the duplication hypothesis, the idea of the heads joined in the middle and the tails at the tip of the arm. The data did not support that. What we found, surprisingly, is the dimension where you cut from the side of the arm to the other side of the arm, so left to right of the arm, that showed the strongest signal for an apiaxis, which was not in any of the morphological models. And it was a big surprise, but it was difficult to really figure out on this technique. We needed more precise localization. So what we did was use this process called in situ hybridization, where we're able to actually label the expression of individual genes, all those important genes that define the head to the tail axis, and look to see where they're expressed in the developing animal. So where we see the pink and the green stain is individual genes labeled up with a fluorescent probe, showing us in what cells they're expressed and in what part of the body plan they're expressed. So this is more easily understood if you think about a mouse. So these are mouse embryos along the bottom here, showing individual genes that define the head. The gene names are not important, but you can see the expression is shown by the expression of blue. So we have a blue dye that we can label up cells that are expressing that particular gene. And you can see all of these different genes have a different position along the developing axis of the brain of the mouse. So this is a technique that in an entire embryo, you can identify the region and the cells that are active and expressing that particular gene along the axis. So what Laurent did was to look at these, first of all, these head genes that define the brain of a vertebrate, and look to see where they're expressed in the developing juvenile of a starfish. And what he found was that all of these genes that define the head are expressed. If you look at the starfish and you flip it upside down so the mouth is in the middle, they're expressed along each of the midlines of the arms. And that is where the nerve cords of the arms extend. So that the most anterior part of the nervous system marker is expressed in the midline of each of the arms. If you look at genes that are expressed in the forebrain, but also in the midbrain, so slightly more posterior in the vertebrate brain, these are now expressed on either side of that midline. So they wrap around the midline on the lateral parts of the arm. And then finally, if you look at genes that are expressed on the boundary between the midbrain and the hindbrain, so where the spinal cord starts in the vertebrates, so slightly more posteriorly, you can see these genes are now expressed wrapped around the side of those developing arms. So it suggests that the starfish is largely on its anterior portion is the whole of the oral surface of the starfish. You flip a starfish upside down, that whole surface corresponds to the vertebrate head, with a midline of each arm representing the most anterior part of the developing brain of a vertebrate. Surprisingly, the genes that define the spinal cord of a vertebrate, the trunk markers that we see in flies and in humans, are completely missing in the ectoderm of the animal, suggesting that as part of the evolution of the echinoderms is they expanded their head, stretched it into each of the arms, but then completely lost their trunk. So they're defined largely by a head territory. And this is sort of diagrammed here in the diagram from the paper that we published. What I want to do is just take the last few minutes. If you didn't understand any of that, and you'd like a slightly more simplistic version, as part of the paper publication, the journal did a beautiful, very short movie explaining the observation, and I'd just like to play that for you.
It's starfish with clothes. Its arms, on all of its arms, on the 2 feet it uses to move. It's an interesting hypothetical, but it's also representative of a pretty fundamental concept in the evolution of animal body plans: bilateral symmetry. That is to say, a body plan with a head end and a tail end with a symmetrical body on either side. Which means for a lot of animals, you could argue that there is a scientifically accurate answer to the trouser question. The trousers go at the bottom or tail end. But starfish, despite being evolved from the same ancestors as other bilaterians, don't seem to have an obvious tail, head, or clear symmetry. Their mouth is on the bottom, their anus is on the top. No clear place to put a pair of trousers. And yet, as larvae, they do have bilateral symmetry. And that is particularly perplexing to developmental biologists. Generally, in the animal kingdom, once you evolve a bilaterally symmetrical body plan, you don't go back. So what's going on? Well, scientists may now have an answer, and they found it by looking at the genes which encode body plans. But before we get to their findings, let's start with the basics. Bilaterial body plans can be roughly divided into 3 sections. 2 of these are in the head: forebrain and midbrain, shown in red and blue. And the third is the rest of the body, the trunk in yellow, at the tail end where the trousers might go. These sections are generally arranged in a straight line along the head to tail axis, and each have different gene expressions associated with them. So how might this map onto a 5-armed starfish? The first theory is a somewhat cartoonish arrangement, with the forebrain in one arm and the trunk opposite. Cute, but previous work has already shown that it's not very likely. Another theory is that each of the starfish's arms is in fact a copy of the basic 3-part body plan, duplicated 5 times and joined at their heads with a trunk on each end. Another popular theory is that perhaps the normal head-to-tail axis goes from mouth to anus, upside down through the centre of the starfish. The forebrain would be at the bottom, then the midbrain, then the trunk on top. To settle the debate, researchers mapped gene expression in different bits of starfish anatomy, looking specifically for the genes expressed in the forebrain, midbrain, and trunk of other bilaterians. And this is what they came up with. The forebrain genes are expressed in the centre, reaching out into each arm. Around that are the midbrain genes. But the trunk is not there. There is a thin layer of tissue that expresses the genes normally found at the boundary between head and trunk, shown here in green. But the trunk itself is absent. The starfish is basically all head. Which means, at odds with all previous theories, this starfish has nowhere to put its trousers. The researchers say that this finding is likely to apply to other echinoderms as well. And it's an important piece of the puzzle when it comes to understanding bilaterial evolution and figuring out how strange creatures like echinoderms relate to creatures like us. Paleontologists also now have more information to go back and study the many strange fossils of echinoderms and their relatives. And in the meantime, instead of a starfish in trousers, perhaps we'll have to settle for a big hat.
So that's basically the story. I'd like to just elaborate on a little bit in terms of thinking about how that might have happened. My daughter did a really good diagram. Completely unbeknownst to me, she handed this to me. She's 9 years old. And her explanation on how to compare a vertebrate to a echinoderm is to chop off the head of a vertebrate, to flatten it down, and to squash the head. I would add that another way to think about it is you then need to take hooks and you need to stretch the brain into 5 different lobes to stretch the brain into the each arm of the starfish. Rather brutal. But my final takeaways is that model species are an essential foundation to our understanding of functional biology and will always remain the point of principle for understanding basic biology. However, the ease at which we can sequence the genomes, and we can now carry out gene manipulators in new species, using and exploiting the wealth of evolutionary experiments that we see in the ocean, offers up a really unprecedented opportunity with these new tools available to us. Animals can be chosen for their unique and fascinating biological properties, such as being able to regenerate their entire body, or being able to resist cancer, or the evolution of unique immunity properties. And my mission at Stanford is to sort of change the nature of part of the Marine Station from being a center of conservation and ecology, which is what most people think of when they think of marine stations. But there's also a place for fundamental science based on biomedical research based out of marine stations. And now is the time to really focus on that mission because of these incredible tool sets that are now available to us that can be applied in new contexts. So with that, I'd like to thank my lab, particularly Laurent Formery, who did the work on the Starfish project, and my collaborator Dan here, and David Rank and Paul Peluso from PacBio. So with that, sorry if I've gone over a little time. All right.
Yeah. Right. So now at Stanford, where you're a professor in marine biology, tell us where you are in the hierarchy. So the overall faculty, with the science, where inside is going to give us a tree.
Where I am in terms of
What I mean is, if you were looking at an organizational chart, a tree of the different departments, where do we get the evolution of biological scientists? All science, and we've probably got bio and mechanical, but tell me the hierarchy.
So I'm in the biology department, which is part of the humanities and sciences. But there's a large part of medical schools, and a lot of my collaborators are actually based in the medical.
Two-ish, 2 million.
Chimpanzees like 5-6 million.
Okay, so 6 million years at the beginning, chimpanzees and back to us, right? So we, that is to say, life itself is a small piece of Earth's history.
Absolutely.
And humans, literally less than a blink of the eye in human history. And so the study of science like this, less than a blink of a blink of a blink of an eye.
Yes.
And so how much of your research now are you using AI in?
So far, we've not used AI much at all. I think that it's buried into a lot of the genomic analyses that we do, but I don't functionally use AI deliberately in any of my work. I think AI is going to become more and more important for, as we dip into animal biology and we sequence new genomes. One of the biggest challenges is identifying all of the genes we're interested in the genome of different animals, and AI is going to play a very large role in identifying new genes in new genomes.
And so the whole study of evolution, I mean, Darwin died, what, 1882 or something like that? So literally not very long ago, 150 years or so ago, Darwin passes away. How was he right about evolution, and how was he wrong about evolution 150 years after his death?
I think the general principles of Darwinian selection have been borne out by many, many different studies. So I think the foundational principles that Darwin came up with have not really been challenged in the new synthesis of developmental biology. There were lots of things he didn't understand. He didn't have access to our understanding of heredity and genomes, right? And so a lot of the principles that we now understand in terms of how biological change happens have really occurred with our understanding of how genomes function and how genes are producing RNA, producing proteins. All of that was invisible to him back in the day.
So now, what was the size of his staff? Darwin's team?
He pretty much worked by himself on the boats, and so pretty much it's humbling to see the kind of contributions that he made as an individual versus many of us with our groups of, I only have 8 in my group, but many labs that have 40 people in the lab, right?
And how many environmental biologists would you think there are in the world right now?
Environmental biologists.
Ballpark number.
I mean, tens of thousands.
Tens of thousands.
Yes.
How many of them are studying the arena that you are right now, which is?
I think the kind of work that we do is a small group of people, largely because most of the money in biomedical science and using those tools is in monospecies. And so it's very much a niche part of molecular biology is mining biodiversity using these tools. Largely it's expensive. And generally the people who are interested in these big evolutionary questions tend to gravitate towards the more sort of organic fields. They're not necessarily well trained in the mechanistic science of molecular biology. And so it's a small group of people who see the connection between insights into big evolutionary questions, people who are willing to go and to get on a boat, go diving, and to root around in the intertidal and understand biodiversity, but also have the skill set to use these molecular tools applied to these interesting questions. So often I find with my students, there are some students who are really good at the bench, who are really good at molecular biology, and there are other ones that are good in the field, who work well with animals, who are good with animal husbandry, but it's the rare student who combines the skill set that's required to make inroads into this field using both field science and molecular biology.
So how much time do you spend in the field and, as you call it, in the bench or in the lab?
So we work on lots of different animals in the lab. And so we have a month that we work at the Marine Biological Laboratory at Woods Hole, where one of the animals we work spawns its embryos, and we work on embryos. And so we work for a month in the field there. I have another project working on regeneration in worms. There are some worms that you can chop off basically their entire body, and they're able to regenerate their entire body. We work in Tahiti at the Gump Biology Station, which is the Gump department store. They own, Mr. Gump bought a plot of land in Moorea right next to Tahiti, and he donated that to UC Berkeley, and it's now a tropical marine biology station. And I go for a month of the year working at that marine station there.
So now you described how bodies are evolving and evolve, and how nervous systems have evolved and evolved. Are there different mechanisms between the way the bodies are evolving and nervous systems are evolving?
Most of our insights into how body plans change are based on the nervous system. It turns out that most of the conservation that we recognized is focused on the nervous system, largely because most people are more interested in nervous system evolution, so there's been more work done on it.
But it seems that the guiding principles of how you organize our nervous system are incredibly well conserved, irrespective of the structure of the nervous system.
Your whole team, how big is it?
8 of us.
8 people.
Yeah.
And how many other nodes like yours do you think exist in your special area of science?
I'd say, in terms of groups that are doing the kinds of work we do in different animal groups, maybe 50 labs total, would you say that? Yeah,
50 labs
50 labs worldwide, yeah.
And what are the universities that are at the top of that hierarchy of scientific research?
I think University of Chicago is a big area of research that have focused efforts to recruit in this area. Harvard, Stanford, Duke. Any of them that have a marine station are often predisposed to try and staff those marine stations with people doing some of this kind of work.
So those of us who have spent time in the Valley and starting companies remember when distributed computing basically took over sort of like the IT side of the world. Until then, we had, you will recall, big mainframes where intelligence was in these hubs where people could study science with supercomputers, Seymour Cray, and those kind of folks were helping us out. But in the era of distributed computing, it turned out that the pipes between the nodes was more important than the size of the hard disk at the end of the nodes. So when I look at octopuses, I wonder to myself, wait a second, they don't have this central nervous system like we do. They seem to have some kind of a distributed intelligence, and worms seem to have some kind of distributed replicable intelligence. Talk to us about these different strategies of what's central versus distributed intelligence in nervous systems.
Most of our fundamental understanding of how brains function are based on Drosophila and vertebrates. Those are the principles that have guided our understanding of how brains function. Now that we started to dig into things like octopus, we started to understand that the fundamental principles that we have defined based on these sort of model species are the only way that you could solve sensory and processing complex information. And so octopus is probably the best understood of the alternative nervous system functions, but most other animals that have a different type of structure of the nervous system, often it's much more distributed. The molecular biology and the functional insights into how nervous system functions have just not been done. There are so few people working on that the kind of lessons we might learn out of a more distributed nervous system is just no one been doing that kind of work. They started to. There's a really interesting guy at Harvard called Nicholas Bologna, who is foundationally trained in mechanistic neuroscience out of model species. He's now launched his lab into 6 different phyla and finding some really cool principles that would not have been predicted based on understanding of central nervous system function for mice and for flies.
So when I was at Electronic Arts, a giant gaming company, we wondered sometimes what was going to happen to the brains of kids who got really, really good at playing these games and having instantaneous responses. We found, decades later that many of them are the best jet pilots that we have in the world. The question is, how is our brain changing evolutionary by what kind of success we have, doing these things with tools now that we're humans? You think human brains are going to keep evolving in different ways where a certain skill benefit, create more offspring for those guys who are good at video gaming?
I think it's very hard to sort of comment on human evolution because the kind of foundational elements of how evolution functions does not function in our current societies. And so whether or not the kind of tasks that we're excelling in is having a fundamental effect on how our morphology and our functions change, I think that's a very hard and almost a more philosophical question to answer.
So nobody could have anticipated the downstream benefits of like the chase for the moon, get on the moon first, landing at the moon, because the technologies that were needed, had to be developed over the next 9 years. But it was quite an amazing benefit that we, especially in the Valley, got to feel as these new technologies found their way to the practical application. Give us some ideas about the practical applications that your research might show us, years or decades later.
So I think that the examples that I've used in sort of introduction is we are sort of confronted with a crisis of trying to tackle pathogens, particularly bacteria. So we base most of our studies on the way that vertebrates have solved adaptability. with antibodies. So most of our drug design is based on our understanding of how vertebrates tackle pathogens. It's very likely from studies that have just sort of gotten started. If you look at all these individual phyla that have had 500 million years of evolution in the ocean, which is jam-packed full of viruses and bacteria, that each of them have solved immunity in a different way, and maybe a fundamentally different way. And by exploring the mechanistic basis of adaptive immunity in different animals, we will have insights into how we can tackle some of the most fundamentally challenging problems to human health, which is the action of pathogens.
So compare the number of species on land with that in the ocean. What's the percentage of life on Earth that's on, in terms of species count, on land and in the oceans?
So that's a difficult metric. So arthropods are by far and away the most speciose phyla on the planet.
Tell people what those are.
Arthropods, so there's flies, beetles, butterflies. The insect diversity is off the charts. The kind of diversity we see in the ocean is not so much that any one phylum is particularly diverse, but that pretty much all phyla are represented in the oceans, whereas on land, only a few phyla are represented on land. Mollusks, like slugs and snails, arthropods, and chordates. Echinoderms are not on land. Jellyfish are not on land. So even though the actual species number may be sort of comparable between the ocean and the terrestrial environment, the representation of biodiversity is by far and away the best represented in the ocean.
So now your whole budget seems amazingly small to me. You said earlier that you guys can live on an annual budget of $500,000 a year or so.
Yeah.
Where do you get your money? Who funds you?
So NSF, National Science Foundation, has been sort of my sort of standard bearer for supporting the work we do. The Starfish project was a NASA-funded project. They have an exobiology program where they try to understand, if we were to identify life on another planet, what are the foundations of life? And I made the argument that exploring axes on our own planet will give us an understanding if there's multicellular life on another planet, it has to be organized in one next. So understanding axis formation is critical. So NASA has funded us. The most important funder of our research for the past 5 years or so, both Dan and I have been the benefits of this, is the Chancellor Zuckerberg Biolab. They have given us money, and basically the most impactful work out of my lab has been funded by very risky projects that were unreservedly supported by Chancellor Zuckerberg. So that has been a really important funding. And then individual groups that give me $50,000, $100,000 on campus, those often are enough to support a nucleating project, which is difficult to get funded by some of the national agencies. So it's a distributed effort, but you have to be pretty nimble to fund this kind of work, because it is not the kind of work that is very easy to write an NIH grant for or an NSF grant for, because generally they want a pretty established and a pretty certain bet to give funding for those kind of projects. Both Dan and I have sort of thrived by being nimble and trying to identify funders who are willing to take a risk on an interesting question.
So do individuals contribute as well, small, normal human beings?
A couple of times people have given me like $10,000 here, $20,000 there, and that really helps funding some field work, for example. There's an identifiable project that we want to buy a piece of equipment for. Right now, one of the projects we're working on is how to carry out single-cell sequencing, single-cell genomics at a dirty, filthy marine station in the middle of the Pacific, where you're trying to do really detailed and complicated genomic work that involves buying a $100,000 piece of equipment, put it in your luggage and take it out to the marine station. Because bioprospecting is going to involve not just working at Stanford University at the marine station, but it's to go into the natural environment to find weird and wonderful creatures that you can only find in certain parts of the world, and be able to equip yourself with the kind of technology that is necessary on a boat to address some of these more fundamental questions.
So my son-in-law is a bioinformatics scientist at Stanford, and he recently was spun off in a venture-backed startup, basically modeling the physical characteristics and chemical, predilections that people might have for certain diseases. And ultimately, some biotech company could buy them, and or a pharmaceutical company could ultimately buy them because they want to use it for diagnostic work and so on. So just imagine somebody who might want to buy your research in the future. Would it be a pharmaceutical company, or what type of company?
Generally, the kind of work that I do doesn't have immediate practical implications. It is foundational science that what one hopes is that the kind of principles that we uncover, the next rung of scientists who's really interested in applying that to a more practical endeavor would see the benefits of the work we do and sort of take it and run it. So I have a hard time thinking about the kind of results that we have discovered, whether or not a company would be interested in sort of buying that technology. We're generally applying novel but established technologies into the interesting biological questions.
So I have a question.
Yes, sure.
I wondered about the starfish. What happens with the rest of the digestive system of the starfish? Does it even have, a stomach or an anus or anything? Just a brain and that's it? Where is the rest of the starfish?
So it's all tucked up inside. So they have a through gut that goes to the mouth, the anus. And it turns out—
Where is the anus?
The anus is at the top. So if you look at the starfish, you look at the top of the starfish, right in the middle of the starfish, you're looking down and it is where the anus is.
But that's where the brain is, no?
That is so the opposite side to the brain.
Turn it over.
Turn it over.
It's on the bottom of the
The
starfish.
brain is, so the way that one of the commentators of the paper said that echinoderms are brains walking on lips, is the way he described them.
Okay, so it's got all that, it's just on the top.
It's sitting on top of the brain. So it's basically the guts and the muscle sits on top of the brain that's walking along the ocean.
And that is that's very good for the starfish to have the brain there.
They seem to have done pretty well they've been a very successful group of animals diversified across all the oceans. So they've done pretty well with that kind of structure.
Yeah, they're a top, aren't they a top? Aren't they the top when they go into the?
So they're a keystone predator in the intertidal here on the west coast. There's the purple starfish and the orange starfish that you see, Physaster gracious, are the defining predator of the intertidal, and it defines the distribution of mussels along the intertidal. So they're an incredibly important part of the ecosystem.
Wonderful. Any other questions from the audience? So, Chris Lowe, marine biologist and exploring the evolutionary development of life on Earth and humans and the biodiversity of all of us. It's fascinating to spend time with you. Thank you very much for sharing your insights with the Wednesday Yachting Luncheon. Thank you, buddy.
Thank you very much. Thank you.